Welcome to this explainer. Today, we're going on an absolutely fascinating journey. We're talking about a concept that literally starts out in the vast, starry expanse of outer space and ends up deep inside the microscopic cellular architecture of the human eye. We're exploring adaptive optics in retinal imaging. It's a stunning story, honestly, of how an engineering marvel designed to look out into the universe was completely flipped to help us look inside ourselves.
Okay, let's dive into this because this is the perfect hook. What could a massive astronomical telescope pointed at the stars possibly have in common with the tools an ophthalmologist uses to look inside your eye? Well, the answer actually lies in the limitations of physics. We often think of space exploration and medical imaging as two entirely different fields. But as we're about to see, they were historically blocked by the exact same visual barrier.
Think about it. If you've ever looked up at the night sky, you've seen stars twinkle. But here's the thing, stars don't actually twinkle. That twinkling effect is caused by atmospheric turbulence. It's basically constantly changing air currents distorting the incoming starlight. Well, a very similar process happens inside the human eye, but instead of atmospheric turbulence, we have what's called ocular aberrations. These are subtle imperfections in your eye that distort the light before it reaches the retina, causing a blurry image. So the exact same physics problem that prevents astronomers from getting a crisp picture of a star also prevents doctors from getting a crisp picture of your retina. Crazy, right?
So here's our roadmap for today's explainer. First, we'll go from stars to retinas. Then we'll tackle the wavefront problem, followed by how we're correcting those imperfections. After that, we'll look at microscale retinal imaging, and finally, wrap up with the clinical impact. Let's get started.
From stars to retinas.
Even in a perfectly healthy eye, light just doesn't travel flawlessly. We're all pretty familiar with lower-order aberrations. These are your standard vision issues like myopia, hyperopia, or astigmatism, the stuff that's easily fixed with a pair of glasses. But the real problem here lies in higher-order aberrations. These are incredibly subtle, complex irregularities hidden deep within the cornea and the lens. And the kicker? Traditional glasses can't fix these. They act just like that turbulent atmosphere we talked about, degrading the light before it ever even has a chance to hit the retina.
Which brings us to part 2, the wavefront problem.
So the crucial point here is understanding this concept of a wavefront. Imagine light traveling as a perfectly flat sheet. In a flawless optical system, that sheet stays perfectly uniform as it passes through the eye. But as we know, human eyes just aren't perfect. Those subtle irregularities warp and distort the wavefront. It's kind of like trying to look at a shiny coin sitting at the bottom of a rippling swimming pool. Because the wavefront becomes distorted, the light simply cannot focus to a single point, and that inherently blurs the image on the retina.
Let's move on to part 3, correcting those imperfections.
This right here is an absolute engineering marvel. It's a brilliant 3-step process. First up, the system has to figure out exactly how the light is warped. It does this using a Hartmann-Shack sensor, which measures the deviations from that ideal flat wavefront we just talked about. Second, a computer instantly analyzes that deviation. And third, this is where the magic literally happens, the computer sends a signal to a set of deformable mirrors. These mirrors physically change their shape in real time to perfectly compensate for the errors. It's constantly flattening the light out before the image is even captured.
Now, just to be scientifically accurate here, adaptive optics doesn't miraculously give us a perfect eye. We're still bound by the laws of physics, unfortunately. Even with this flawless, real-time correction, things like intraocular light scatter and neural visual processing still get in the way. And for patients with small pupils, diffraction is actually the main limiting factor, not aberrations. But make no mistake, adaptive optics successfully removes the single biggest historical roadblock to clear imaging.
That unlocks part 4, microscale retinal imaging.
Because we've removed those pesky higher-order aberrations, something truly spectacular happens. We can stop looking at the eye as this generalized structure and start looking at it as a microscopic universe. Now, what's really interesting about this comparison is the dramatic contrast in the data. We're looking at optical coherence tomography, or OCT. Now, conventional OCT is already fantastic when it comes to depth or axial resolution. Both are under 3 micrometers. But look at the lateral resolution. Standard imaging is totally bogged down by those ocular aberrations, limiting us to around 15 to 20 micrometers. But when we integrate adaptive optics to correct those aberrations in real time, that lateral resolution plummets to an incredible 2 to 3 micrometers.
2 to 3 micrometers. I really want to emphasize that number for a second, because it is literally the threshold of a medical revolution. When you improve lateral resolution to 2 or 3 micrometers, you're no longer just looking at blurry layers of tissue. You've crossed the threshold that actually allows you to distinguish individual retinal cells in a living human eye. It's mind-blowing.
Because of this massive leap in resolution, our images correspond incredibly closely with actual retinal histology. We can identify the microscopic architecture just based on how different structures reflect light. For example, notice how the top retinal nerve fiber layer is highly reflective. Then, deeper down, foundational layers like the retinal pigment epithelium and Bruch's membrane also bounce back strong reflective signals. Meanwhile, the vitreous fluid appears completely non-reflective. Adaptive optics maps all of these varying signals into a crisp, microscopic landscape.
And it gets even better when we start combining technologies. Adaptive optics is often paired with scanning laser ophthalmoscopy, or SLO. SLO already creates really high-resolution images by scanning the retina point by point with a laser. It uses confocal technology to accept specific, focused light while rejecting all that scattered, blurry light. So, when you add adaptive optics into the mix to strip away those aberrations first, the result is this absolutely unprecedented, highly detailed 3D view of the retina's microstructure.
Finally, let's talk about Part 5, the clinical impact.
At the end of the day, the true value of adaptive optics is what these images reveal for actual patients. By allowing clinicians to study microscopic architecture in vivo, which means in a living, breathing person, rather than on a lab slide after the fact, we are completely changing the game. A prime example here is glaucoma research. Thanks to this tech, we can now visualize microscopic lamina cribrospores, connective tissue beams, and even individual nerve fiber bundles. Seeing these structures opens the door to the absolute earliest possible disease detection.
And this brilliantly illustrates our core takeaway for today. Adaptive optics represents an incredibly important bridge between optical physics and clinical ophthalmology. We're witnessing a real convergence here. Modern imaging is increasingly layering these technologies together, from various forms of OCT to confocal SLO and adaptive optics, all working in perfect harmony. And while this tech is currently concentrated mostly in research centers, the instrumentation is becoming faster, it's becoming easier to use, and it is rapidly moving toward routine clinical practice. Which leaves us with this final thought to ponder. We started out today looking at how astronomers correct the twinkling of stars, and we ended up visualizing individual living cells inside the human retina. We've literally removed the barriers of optical aberrations to unlock cellular-level pathology in real time. So I'll leave you with this. If our technology has evolved to the point where we can now monitor the human eye cell by cell, what hidden microscopic signs of disease will we uncover and ultimately cure tomorrow? Thank you so much for joining me on this explainer. Keep learning, and I'll catch you next time.